Hello, I'm Sudeshna Das, an assistant professor of neurology at Mass General Hospital and Harvard Medical School. It's my distinct pleasure to speak at the 35th anniversary of iOS Press. I sincerely thank Dr. Einar Fredriksson and colleagues for giving me this wonderful opportunity. Today, I'll be presenting Alzheimer's DataLENS, an Omics portal for Alzheimer's disease research. First, I'll introduce DataLENS, then give a brief summary of current status, followed by a preview of DataLENS 2.0, our next generation of the portal, and finally end with conclusions. So what is DataLENS? DataLENS is an open data analytic platform that aims to advance research in Alzheimer's disease and related dementias by making omics data available to all neuroscientists. It provides consistent pipelines to process and analyze omics data from different sources. It has easy-to-use web interfaces for query and visualization of all the analytics. It integrates information from multiple heterogeneous modalities to present an integrated view of molecular mechanisms to a neuroscientist interested in Alzheimer's disease research. We house different kinds of data in DataLENS. Currently, the primary source of data is gene expression data. We have analysis of over 30 public human data sets spanning 19 brain regions and 5 cohorts. We also include various animal models of Alzheimer's disease, as well as data sets from other neurodegenerative diseases such as Lewy body disease and ALS-FTD. We've also started including data from new technologies such as single nucleus RNA-seq, and we have 16 different query and visualizations tools available to the neuroscientist for browsing and viewing this data. In addition to gene expression data, we also include proteomics data, GWAS studies, and pathways data as well. Here is a screenshot of the homepage of the current version of DataLENS. DataLENS was created with seed funding from the MassCats project, which is a public-private partnership to discover new treatments for Alzheimer's disease in the Boston area. DataLENS has resulted in multiple publications, ranging from understanding neuronal calcineurin transcriptional targets in both mouse models and Alzheimer's disease brains, to understanding angiogenesis-related gene expression, to studying the microglial response to A-beta plaques, to performing meta-analysis of mouse transcriptomics studies, to understand astrocyte reaction, to characterizing clinical and neuropathological traits of ApoE haplotypes. So in summary, DataLENS has provided a large amount of research so far. DataLENS currently is hosted on Drupal, which is an open source content management system. It is built on Drupal 7, and the current version of Drupal is Drupal 9, so it's been outdated. We plan to develop DataLENS 2.0 using state-of-the-art tools such as RShiny with an interactive HTML5 interface. These DataLENS is hosted on our institutional servers and maintained by our IT staff. We plan to host it on the Amazon Web Services and make it an open platform. Currently, it can hold only small data sets, but with the advent of new omics technologies, we have to scale it to be able to handle large data sets and make sure that the performance is maintained, and we can do queries on large data sets in a quick and flexible way. Next, I'm going to show a prototype of DataLENS 2.0 and walk you through an example exploration of single nucleus RNA-seq data. So here's a preview of Alzheimer's DataLENS 2.0, where we can query gene expression data, differential gene expression analysis from both bulk gene expression and single nuclear RNA-seq data as well. Here is a little preview of the navigation bar where users can explore the different kinds of analysis that will be available in DataLENS 2.0. We also are planning to provide wiki pages so users can understand how to get started by querying for gene expression data, or even understand the overview of how single nucleus RNA-seq data was done, and perform other kinds of analysis, such as looking at cell proportion data or cell type clustering data as well. Next, I would like to walk you through how to query cell-level gene expression data. We'll make it really easy for the user, where they can just type in their gene name of interest, choose how to explore the data with various different traits, such as BRCA or SIRAD neuropathology or ApoE genotype, choose the type of plots that they're interested in, select the data sets, as well as able to download the visualization and plots for their manuscript if needed. This video just illustrates how someone can look for ApoE across various CRAD stages, for example, and then look at the data and various brain regions, and then finally be able to download that data. Next, I'd like to show how we can use DataLENS 2.0 to query the average gene expression of a particular gene across various pathology and data sets. So here is an example of sample level data across 3 different cell types: neurons, astrocytes, and microglia, in 4 publicly available Alzheimer's disease single nucleus data sets from 4 different labs. So for each of the cell types, the expression for a particular gene are shown across 3 BRCA stages of low, medium, and high for these 4 particular data sets. One can perform a similar query of average expression of gene set of interest, as well as a particular gene level. So here you would take the average expression of all the genes rather than one single gene, and again have a similar visualization across multiple cell types and data sets. This is an example of the same that we have done, and show how this whole query is interactive, and also once the plot is created for ApoE gene expression across CRAD, for example, and multiple data sets and brain regions, one can download the data for publication quality as well. Next, I'd like to show how users can query for cell type proportions, and they can choose to look at the cell type proportions at the level of each donor or each sample, or aggregate it with pathology such as BRCA or SIRAD. Choose the type of plot, either a box plot or a bar plot, select the various data sets or the cell types of interest for querying the cell proportions of, and then finally, again, any visualization that is created in DataLENS, the users will be able to click a download button for looking. And then finally, I would also like to show how users can look at how the clusters from one data sets relate to others. So here's an example heat map of correlation analysis that was done across different cell types, subclusters from different data sets. And here one can look at, is the data set that was derived from the entorhinal cortex is similar to that from another region such as the dorsolateral prefrontal cortex, and again, this whole visualization, we plan to make it interactive for the users to investigate and explore. So the heat map is also interactive. So for example, one can choose a certain type of clusters, zoom in on that data and look at it in further detail. Again, it's the same theme of interactivity for the user to be able to look at multiple different data sets, large omics data sets, and explore it with easy to query user interfaces with simple clicks and searches for the user. And again, any analysis or plots that are created in DataLENS, we plan to make them downloadable for publication purposes or other presentations as well. Here, I'd like to switch gear and talk about the Astrocyte Atlas, which is a public compendium of astrocyte IHC data from postmortem Alzheimer's disease brains. Again, one can look at the network analysis of this data, for example, and one can select the gene of interest and zoom in and into an area of interest. Again, I would like to highlight the theme of interactivity and simplicity in exploring all of this publicly available data. So here's another example where we perform transcription factor enrichment analysis on this data set, and one can explore this data as well by zooming in on the different transcription factors that may be responsible for Alzheimer, for astrocyte reaction in Alzheimer's disease research. DataLENS is an open data analytic platform that aims to advance research in Alzheimer's disease and related dementias. DataLENS is an easy-to-use interactive query and visualization portal for ADRE researchers that integrates data from multiple public resources. We hope to continue development of the next generation of the portal, that is DataLENS 2.0, with support from iOS Press, and more importantly, in collaboration with the whole JAD community. Finally, I would like to thank the entire DataLENS team, in particular Xiaoji Li, who is a bioinformatics analyst in my lab, Emily Merrill, Aziz Mezlini, Nate Billmeyer, Max Levinson, Tim Clark, and last but not the least, Dr. Brad Hyman, who is the principal investigator of MassCats, who provided seed funding for this project. Thank you all for listening.